Welcome to Endo the Cell, the podcast where we shrink ourselves down to cell size and investigate the microscopic mysteries of life. No lab coat required. I'm Amanda.
And I'm Josie. Today, we're talking about how cells eat. But don't picture a cell with a tiny fork and knife. We're talking about endocytosis, the process where cells pull in nutrients, molecules, and signals from outside.
Yeah, you could say it's a cell's version of shopping on Amazon. It orders molecules and then ships them right into its cytoplasm.
2-day delivery, you could say Amazon Prime, courtesy of the plasma membrane.
Exactly. But here's the weird thing. Scientists didn't actually know how the cell physically pulled this off.
Yeah, imagine trying to take a floppy fluid membrane and then fold it in a little bubble and pinch a bubble of it off. It's like folding soup. So the cell is basically out here doing the impossible, folding liquid into more liquid.
That's a great way to imagine it, Josie. Pretty much impossible without some serious muscle. And that led to one of the big questions in cell biology. What's the engine behind endocytosis?
That's such an interesting question, Amanda, because the membrane doesn't just magically fold, it takes force. So what's powering that inward pull?
Enter actin, the unsung hero of the cytoskeleton.
Actin really runs the ship. Actin's the kind of protein that's everywhere. It builds structure, moves cells, helps them divide, and apparently handles snack time too.
That's a good one, Josie. For years, scientists suspected actin might be involved in endocytosis, but they couldn't prove it. It was like having a suspect, but no video evidence.
But then, Pia Kaxinen and John R. Durbin decided to play cell detectives.
CSI Cytoplasm Edition.
Exactly. Around 2005, while working at UC Berkeley, they used live cell fluorescence microscopy to watch yeast cells undergo endocytosis in real time.
Which honestly is brilliant. Yeast cells are simple, fast-growing, and perfect for this kind of work.
They tagged the proteins involved in endocytosis with fluorescent markers so that they could literally watch the process unfold, like a neon-colored light show happening inside of a cell.
So they could actually see when actin appeared.
Right. And what they found was incredible. Actin didn't hang around the whole time. It swooped in right before the vesicle separated from the membrane.
So like the friend who shows up at the exact moment you need help moving that heavy couch.
Exactly. This was the burst of actin polymerization, a rapid buildup of filaments that generated the force to push the membrane inward and to push off the vesicle.
So actin wasn't just helping, it was the driving force, the engine of endocytosis.
It's like the cell's own rocket booster system. Just a quick push and whoosh, the vesicle launches into the cytoplasm.
And once the vesicle forms, the actin filaments disassemble. Mission complete.
Talk about efficiency. It's the mission impossible of the cellular world.
And this discovery totally changed the way scientists understood endocytosis. Before Kaxinen and Durbin, people thought it was mostly passive, like the membrane just folded in on its own.
But now we know it's an active, force-driven event powered by actin dynamics, meaning the cell puts in energy, timing, and coordination to make it happen.
And that has huge implications beyond basic biology.
For one, endocytosis is how cells take in drugs and nutrients. So if we understand how actin controls that process, we can design better drug delivery systems, basically using the cell's own machinery to get the medicine exactly where it needs to go.
Or on the flip side, we can figure out how to block viruses that use the same trick to sneak inside.
Right. Viruses are like professional hackers of the cellular world. They hijack endocytosis to break into the cell, and actin is the getaway car.
So by studying these tiny molecular forces, we're not just learning how the cell eats, we're uncovering how diseases invade and how treatments can outsmart them.
And all thanks to Kaxinen and Durbin, who basically gave us front row seats to the cell's private performance.
Honestly, it's wild how something so microscopic can reshape medicine and molecular biology.
Makes you appreciate your actin filaments a bit more, huh?
Yeah, definitely. Thanks for holding me together, actin.
Huge shout out to Pia Kaxinen and John Durbin, the dynamic duo who showed us that actin isn't just structural, it's powerful.
And if you like today's episode of Endo the Cell, give us a follow and share it with your lab partner, biology buddy, or that one person who still calls the mitochondria the powerhouse of the cell.
Next time, we'll zoom in on another jaw dropping discovery from the world of cell biology.
Until then, stay curious, stay caffeinated, and don't forget to endo the cell.